Introduction. Welcome to today's lecture on cell biology, specifically focusing on the fascinating field of hematopoiesis and hematopoietic stem cells. Hematopoiesis is the process by which all mature blood cells are produced, and these cells originate from a rare population of cells called hematopoietic stem cells, HSCs. HSCs have the remarkable ability to self-renew and differentiate into various types of blood cells, making them vital for the maintenance of the hematopoietic system throughout our lives. Now, let's delve into the intricate details of hematopoiesis and explore various aspects related to HSC maintenance, lineage commitment, transplantation, aging, diseases, and therapeutic applications. Hematopoietic stem cells reside in specialized niches in the bone marrow microenvironment. Various cell types secrete regulatory factors that help maintain and renew these stem cells. Signaling pathways and transcription factors play essential roles in regulating their fate, along with epigenetic modifications. Understanding these mechanisms is vital for regenerative medicine and treating hematopoietic disorders. In the process of blood cell formation, certain factors and modifications control the specialization of hematopoietic stem cells. Transcription factors like GATA1, PU.1, and C/EBP-alpha activate specific genes and suppress alternative genes. Epigenetic modifications, such as DNA methylation, histone modifications, and chromatin remodeling, also contribute by regulating the expression of lineage-specific genes. Studying these mechanisms is vital for understanding blood disorders and discovering potential treatments. Hematopoietic stem cell transplantation, HSCT, is a game-changing treatment for blood disorders and genetic diseases. It involves replacing the patient's diseased cells with healthy stem cells from a donor. Gene therapy is also showing promise in correcting genetic defects before transplantation, offering potential cures for many diseases. The bone marrow microenvironment is vital for hematopoietic stem cell, HSC, regulation. Osteoblasts provide physical support and secrete factors like stem cell factor and N-cadherin. Endothelial cells secrete angiopoietin-1, promoting HSC attachment. Immune cells modulate HSC regulation. Studying the HSC niche is important for transplantation and regenerative medicine advancements. As we age, hematopoietic stem cells undergo changes that can affect their function and regenerative potential. Accumulating evidence suggests that aged HSCs exhibit functional decline, decreased self-renewal capacity, and skewed lineage differentiation. Various intrinsic and extrinsic factors contribute to the aging process in HSCs. Intrinsic factors include alterations in gene expression, increased DNA damage, and telomere shortening, while extrinsic factors include chronic inflammation and changes in the bone marrow microenvironment. Understanding the molecular mechanisms underlying HSC aging is crucial, as age-related changes in HSCs can impact overall blood cell production and potentially contribute to the development of hematopoietic disorders associated with aging. In summary, hematopoietic malignancies, such as leukemia, result from abnormal hematopoietic stem or progenitor cells. Leukemogenesis involves genetic and epigenetic changes that disrupt blood cell development. Understanding these alterations and developing targeted therapies are critical for improving leukemia prognosis and treatment. Bone marrow failure syndromes, including aplastic anemia, are characterized by a decrease in the production of blood cells, resulting in low blood cell counts and a compromised immune system. Aplastic anemia is primarily caused by immune-mediated destruction of hematopoietic stem and progenitor cells, although other factors, such as genetic mutations and exposure to certain environmental toxins, can also contribute to its development. Understanding the pathogenesis of bone marrow failure syndromes is crucial for developing targeted therapies to enhance hematopoietic recovery and improve patient outcomes. In the video on hematopoiesis and blood cell formation, we will explore how different waves of hematopoiesis occur during embryonic development. We will discuss the complex interplay of transcription factors, cytokines, and signaling pathways that orchestrate this process. We will also examine the formation of primitive erythrocytes in the yolk sac and the emergence of hematopoietic stem cells in the fetal liver, which give rise to definitive blood cell lineages. Understanding these mechanisms is crucial for regenerative medicine applications. Hematopoietic stem cells have great potential for regenerative medicine. They can renew themselves and form different blood cell types. We can transplant these cells to treat diseases like heart conditions, immune deficiencies, and some cancers. Challenges like cell engraftment, immune rejection, and safety concerns still need to be addressed. For successful transplantation or therapeutic purposes, hematopoietic stem cells must be mobilized from their niches within the bone marrow and directed to the desired sites. Mobilization involves the release of HSCs into the bloodstream, where they can be harvested for transplantation. Numerous factors, including cytokines such as granulocyte colony stimulating factor, G-CSF, chemokines, and adhesion molecules, play a role in regulating HSC mobilization and homing. These factors act collectively to influence the migration, adhesion, and extravasation of HSCs. Understanding the intricate mechanisms of HSC mobilization and homing is essential for optimizing transplantation procedures and developing strategies to enhance stem cell engraftment. Hematopoietic stem cell fate determination and lineage specification involve the activation and repression of specific genes through transcription factors, epigenetic modifications, and signaling pathways. Examples include GATA1 and PU.1 driving erythroid and myeloid lineage commitment, respectively. Specific enhancers with epigenetic modifications regulate lineage-specific genes. This knowledge is important for understanding hematopoietic disorders and developing targeted treatments. Hematopoiesis and blood cell formation are closely connected to the immune response and inflammation. Infections or injuries trigger an increased production of specific blood cell lineages for an effective immune response. Inflammatory signals directly affect stem and progenitor cells, influencing their growth, specialization, and movement. Chronic inflammation can disrupt blood cell production and increase the likelihood of hematopoietic disorders. Studying the relationship between hematopoiesis and the immune response is vital for understanding inflammation-related hematopoietic disorders. In conclusion, hematopoiesis and blood cell formation are crucial for a healthy blood system. The balance between stem cell renewal and lineage commitment is controlled by complex factors. Studying these mechanisms can lead to advancements in research and medical treatments for blood disorders. Thank you for joining me on this journey.